Hello, hello. Time to talk about Chapter 5. The way I'm going to approach this is I'm going to talk about the first part of the chapter, which details the structure of the cell membrane. But in terms of the actual transport processes, instead of putting that in a presentation here, an annotated PowerPoint, I've put several links, a link to each type of transport process on the course sites folder for Chapter 5. And each one of them is an animation, okay? And so between your book and the animation, they should make them very clear. They're not hard. It's just that it's you got to keep several of them clear. So I would watch the animations. They're very, very helpful. Okay, let's move on. So again, the phospholipids. I know we've talked about the phospholipids and the fact that they make up the bulk of the membrane. But as you're reading in the book, this is one of the four primary components. And you notice that you've got this choline component and this phosphate component, okay, and then of course the glycerol and the fatty acids. So all of this together comes together to make a phospholipid. And again, everything out here, if I change the color here to, well, let's go orange just for fun, everything sticking out here, this part of it, which represents all this stuff up here, is going to be polar. Okay, again, that represents this circle component. And the polar part is going to be hydrophilic. It sticks out of the membrane, whereas, change the color again, let's go yellow maybe, all of this stuff, which equals this stuff, which equals this stuff, all right, that stuff is hydrophobic, and that is non-polar. And that's the stuff that orients in towards the center of the membrane. These are the fatty acid chains, right, composed of carbon and hydrogen. And so these things will self-orient into a lipid bilayer. And the lipid bilayer, again, remember, it is fluid, okay, meaning things move back and forth as we saw in those videos. Okay, so we look at a membrane and we see that there are lots and lots and lots of components to membranes, okay? And this represents the different types of things that we see there. The transmembrane proteins here, and integral proteins, these are the proteins here that are going to go, provide channel access, or as we see here, provide anchoring points for the cytoskeleton, okay? The intermediate filaments here. Here's some actin zipping around there. So we have anchoring points, peripheral proteins, as you can see, they're again served as anchoring points. There's the actin molecule. Okay, other components here, two classes of molecules that serve as basically name tags. The first one are going to be the glycoproteins. Okay, and here is one that's attached to the cell surface, and here's one that's up here on the extracellular matrix, meaning outside the cell. These feathery things are just chains of carbohydrates, okay, on the protein. But we can see these chains here, the individual hexagons of glucose here. And these glycoproteins, and over here you have the glycolipid. The only difference is here the glycogen, meaning the chains of these glucose molecules, are attached to a protein molecule, an integral protein. And on the glycolipid, they're attached to the surface of one of the phospholipids. Both of them, in various circumstances, are going to serve as name tags, either for the tissue types or for what we call the major histocompatibility complex. Essentially, the immune system will recognize cells with the right name tag and it won't kill them, versus cells that don't have the right name tag will get killed. Okay, examples. But it's name tags, so that the body knows that the cells that are in there, as long as they're expressing the right chemical name tag, they're supposed to be there. And of course we see our little buddy, I'm going to change the color to yellow, see our little buddy cholesterol, always cholesterol, assisting in keeping the membrane nice and fluid. Okay, this table in Chapter 5 is very, very useful because it goes over the four major components of the membrane, from the phospholipid structure to the transmembrane proteins. Essentially these are your channels, the interior proteins determining shape and attachment points, and cell surface markers, the glycoproteins and glycolipids, which we talked about, show the essentially the name tag function. So it would be a good idea, again, just to know what's the difference between these four major classes okay, 1, 2, 3, and 4 of structural components to the membrane and where you find, each one. So if I said carriers or channel proteins are part of which class of proteins, you would say, oh, number 2, transmembrane proteins. If I said that, a cell surface marker that determines, like the name tag of the cell, what class of molecule will that be? You say, well, that's cell surface marker with glycoproteins or glycolipids. All right. Okay, so we have the different functions of proteins here. All right, so there are several protein functions. And we have the 6 listed here. So you can be a transporter. This is the one where we talked about. For example, this one here looks like it might be a facilitated diffusion transporter. Molecules going outside in or inside out. An enzyme. You can have an enzyme embedded within the membrane that's going to bind to a certain product and release another product. Okay, and oftentimes this will be useful for molecules on the interior subsurface of the cell membrane, but they can be enzymes. Cell surface receptors. If you get a molecule from outside the cell binding to a protein that sticks on the outside, it will cause a change to the inside of the cell, and this little radar thingy here is meant to say, sends a signal inside the cell to cause something to happen. So it's like putting a key in a lock on the outside of the cell and causing something to happen on the inside. Very, very common. Oftentimes accompanied by G protein receptors. Won't have to get into that just now, but this is a very, very common structure. The cell surface identity markers. Here are your, this one here would be a glycoprotein, but again, you could have a glycolipid off of the lipid molecules right there, the phospholipids. So cell surface marker, but we're talking about proteins, so this one's a glycoprotein. Cell-to-cell adhesion. Again, you essentially have, when cells need to have tight connections to each other, you link them together with proteins. Okay, it's oftentimes also how, for example, an immune system cell will know to glom on to a foreign cell in order to kill it, because there will be this recognition feature with the protein markers, and it'll grab on with proteins. And then attachment to the cytoskeleton. Okay, you have this, this peripheral protein right there, and here you have your actin filaments, and it provides one of those anchoring points for that vast network of the cytoskeleton. Okay, this is just showing that, yes, you can anchor proteins directly to a phospholipid. Remember, the top of a phospholipid, okay, this is polar, okay, composed of choline and phosphate. Okay, when we have that structure, and there's a lot of negative and positive charges up there that you can attach things to, and so you can form this attachment to a protein, all right, and anchor it directly to the surface of the membrane. That's all that's showing. Okay, here's a nice representation of domains, all right. So we have here a couple of examples of transmembrane proteins. This one here, again, you're going to have, and I'll do it in the regions that will be hydrophilic. So we have these hydrophilic regions here in the areas exposed to water in both instances. And then, do it in yellow, we have the hydrophobic regions that will associate inside. And these would be this is a great representation of domains. Okay, so remember, domains in proteins are these regions. Okay, so this would be the hydrophobic domains. And then here would be the hydrophilic domains. Okay, and in both cases here we see a bunch of alpha helices, these helical structures. In the one on the right, it's just one alpha helix, so you might have maybe a receptor protein or something like that. In the one on the left, since you have a lot of different alpha helices, this might be a pore protein, a transmembrane protein that would be a channel, because all those alpha helices form a circle and maybe open up a channel for something to go through from the outside to the in or inside to the out. Okay. Again, here's another example. Okay, this is the retinal chromophore. Okay, where you have retinal, and this is retinal here. And retinal is just a version of vitamin A. Okay, and I believe this is the bacterial retinal chromophore, and it responds to photons of light. Okay, and you can have this complexing deal happening within the protein that is integral in the membrane. So there's a lot of different structures that can be created through the coordination of molecules with these proteins. This is very similar to what happens in your eye in the retina, okay, the retina. In the retina, we also have the mammalian retinal, and it is lodged in a protein, and it responds to photons of light, and it is actually what allows photons of light coming into our eye to send signals through our nervous system to our brains. Okay, beta-pleated sheets, this is just another example. Again, you have this great big porin protein here, forms a big open hole in the cell membrane with all these, they call them anti-parallel, some going this way, some going this way, and it forms this mesh that jams in the membrane and allows there to be a giant pore that opens up in the membrane. So other than alpha helices, you have those beta-pleated sheets. That is a motif, and this domain crossing the membrane, just more structures of proteins in the membrane and the different structures they can take. And this is just to show you what we mean by that trickery we talked about when you were doing your starch and iodine lab. If you notice here, you have a semi-permeable membrane. When you have, okay, solutes on one side of the membrane, the water molecules will gather around them. Okay, they'll bunch up around them, and because the water molecules are no longer freely movable, they've been bound due to their charge around these ions that have stuck in the water. The water molecules on the right side see that, and they say, oh, there are not very many water molecules on the left, because it looks like there are not. There are only a few circulating because all the other ones have been bound up to the solute molecules. So it means that the free water molecules are free to pass this way and keep doing so as long as there is solute on the left for molecules of water to bind around. This was analogous to saying, oh, the iodine moves in, okay, and binds to the starch, but because it binds to the starch, the iodine on the outside of the bag doesn't see the free iodine anymore, even though there's iodine in there, because it's all bound up with the starch, so more and more and more moves in, which is why all the iodine in your liquid moved into the bag and complexed with the starch. Okay, so I'm going to stop there, and again, the rest of it is on the specific means of transport across membranes, including passive transport like facilitated diffusion and osmosis, and active transport and secondary active transport, and exocytosis and endocytosis. And again, I have those all linked with individual animations on the course site's website next to this link for the video. Okay, so just again, knowing the structure of the membrane and then those ways of transporting across.